Whew. G'day everyone. Yes. Points. Leslie, Sophia, points. Everyone else? Welcome to the closing ceremony. And we're going to kick off with our 10th and final plenary presentation. Some 25 years ago, when I was at my very first SMM conference in Maui, there was a gathering of biologists in front of the conference venue one spectacular evening. There may have been some friendly banter going on amongst the rest of the crowd, in which I asked the question, like people and their pets that end up converging on the same phenotype, could it be that senior biologists end up looking like their primary study subjects? I wasn't necessarily looking at Mark, but he said to me, "Simon, that is the end of your career in marine mammal science." We were both wrong. Mark does not have a massive nose, and nor is he 2,000 kilograms. And I'm still here. Mark has made more than 25 research trips to the Antarctic and sub-Antarctic. He's supervised 50 PhD students and worked with numerous international programs, including being the founding chair of the Scientific Committee for Antarctic Research Expert Group on Birds and Mammals. To bookend our conference with another luminary in marine mammal science from Australia. Mark is from the University of Tasmania, and the title of his final plenary of SMM 2024 is Conservation and Management of Marine Mammals, Lessons from the Southern Hemisphere. Mark.
Thank you, Simon, and thank you everybody for being here. The last talk on the last day is always a bit of a graveyard shift, but it's fantastic to see so many of you here, so thank you. Even if, and spoiler alert, it's going to be quite a bit about seals in this talk, not so much about whales, so. Hey! Excellent. Got the right crowd in. So, Simon was just reflecting on his first marine mammal conference. Well, my first marine mammal conference was in 1989 in California. I still remember it vividly. It was a really exciting time for a young PhD student. I got to meet some of the true legends of the society and found out that by and large they were normal people. I got to meet a lot of new friends, some of which I'm very happy to say are still friends today. I learned some really valuable lessons, one of which I should probably share with you, and that is do not go out drinking with Nick Gales and Rob Harcourt the night before you give an important talk. It's a very bad idea. So, it's been fantastic this week to come back to a marine mammal meeting and see that that same sense of excitement is still there. You can tell that people are really invested in the science, they're enjoying the networking, and it reminds me just how important these meetings are as part of our scientific lives. They're really fundamentally important, and this one has been a complete cracker. So, Simon, thank you. Thanks to the committee for organising such a great meeting. Right, let's get the talk going, I guess. We're going to move our focus to the Southern Ocean. So if you were to jump on a boat here in Perth and point it south and head for Antarctica, it would probably take you about a week to get there. It's a long way away. And on the way down there, you'd cross a couple of quite significant oceanographic boundaries, the Antarctic Polar Front, the Antarctic Circumpolar Current, both of which act to really isolate the Antarctic ecosystem from the rest of the world. There's very little exchange between the Southern Ocean ecosystems and those further north. And you know, the whales that we've heard so much about this week are probably a major exception to that rule, but by and large, they're quite isolated. It's also a pretty tough place to be if you're an organism, because it's, well, I don't need to tell you, it's cold. It's really cold. I mean, the oceans freeze, for goodness sake. It's a very cold place. It's so cold that on the Antarctic continent itself, there's almost no life. There's a few invertebrates, a few mosses, but it's effectively barren. And I can say that because I'm quite confident there are no botanists in the audience who are going to give me a hard time. But it's the oceans that really come alive in the Southern Ocean. They teem with life. There's an enormous diversity of organisms that have become adapted to living in this really challenging environment. They live in complicated, intriguing ecosystems, and it's just a wonderful place to visit and to be an Antarctic, to be a biologist. So that's the good news. The bad news is that even though it's isolated, it's not so isolated that it doesn't have its own conservation and management problems. It has actually quite a few. It's quite a big topic to cover, and so I've chosen, rather than try and cover off on all of it, I've chosen instead to take a unashamed personal approach to what I'm going to do. I'm going to talk through some of my own research that I've done over the years, focus on 3 or 4 aspects of it, and hopefully in doing that I can tease out some important broader lessons in terms of conservation and management of marine mammals and what we can learn from the Antarctic. So my personal journey started almost exactly 40 years ago, when I arrived at Macquarie Island as a fresh-faced, newly kitted out employee from the Australian Antarctic Division. I went to Macquarie Island for a year, a bit more than a year in fact, and my job wasn't very challenging. My job was to count things. In fact, I had to count seals. There had been some reports from other islands in the sub-Antarctic that elephant seal numbers were going down, and we were interested to see what was happening with the seals at Macquarie. So this is a photo of me on my very first day at Macquarie Island with my obscenely shiny red coat, talking to one of those animals which I've had a lifelong fascination with ever since. And why wouldn't you? I mean, look at that face. You've got to love those little guys. So I spent a year there. I went out every day and I walked around the beaches and I counted the seals on the beach. And when I came back, I scoured all the records that we had at the Antarctic Division to try and find out what the population used to be. Because nobody had done that. And this is what we found. We found that between 1948, when the first Australian Antarctic expedition went to Macquarie Island, and to 1985, when I got there, the population had declined substantially. It declined by as much as 50% over that 40-year period. And I mean, that was a real surprise, because there's been people on the island continuously during that time, but the rate of decline was very small. It was about 2% per year, and nobody had picked it up. So we were able to document a substantial decline in the occurrence of what is really an important Southern Ocean predator. So in some ways, I was sort of thrown in the deep end when it came to management and conservation issues, because the very first thing I did was to uncover a substantial decline in an important predator. Just to put this in context, what was also happening about that time was that there was growing interest in the idea of climate change, and at the time I was there were reports coming in about glacial retreat on Heard Island and in various other places. But also there are increasing concerns about fisheries in the Southern Ocean. And an organisation called CAMELO, which stands for the Commission for the Conservation of Antarctic Marine Living Resources, was set up about this time to try and keep some sensible management of Antarctic marine living resources. So there's been a lot of talks at this meeting about using marine mammals as sentinels of change. And I think this is an early example of where we've got a top predator whose numbers have changed significantly over time. So it probably fits that criterion of being a sentinel species, although I have to say, full disclosure, I actually am not particularly fond of that concept, at least when it comes to these guys, and I'll talk about that a little bit further on. So this is where we come to the first of my lessons, and that lesson is we just don't know enough. Back in 1984, we certainly didn't know enough. We had no idea what was driving this decline. We knew things that we didn't know, the old known unknowns. We had no idea about the basic demography of these guys, who was living and who was dying. We had no idea where they went, and we had no idea of what they were eating. And I guess particularly important in the context of my earlier statements, we had no idea how those things related to the marine resources. So it was pretty much a blank slate. So we then spent the next 40 years trying to answer these questions. So I'm just going to quickly touch on some of the things that we've found since then. It's not a comprehensive analysis or review of all the work that we've done, so apologies to folks that have done stuff that I don't mention. I'm really just trying to pick out a few things that will give us a bit of a narrative here. And the first thing was demography. Now, we were lucky that the early biologists at Macquarie Island in the 50s and 60s conducted a big demographic program. They branded many thousands of pups, and they matured and moved into the adult population. In fact, the photo there, if you look carefully, you can see that animal's got a brand on her back. That was an animal we saw in 1985 who'd been branded back in the 1960s. So we had that data to draw on, and I'd just like to acknowledge Harry Burton, who was really significant in a lot of the work that we're doing here. He instituted the long-term elephant seal study through the Antarctic Division, which really was the basis of everything that I'm going to be talking about for the next few minutes. So what Harry did was institute a new branding program in the 90s so that we could get some information about these really key demographic parameters that were driving the population. And there are a couple of things that really stood out, and one of them is this intriguing figure here, where we can see that juvenile survival, so that's first-year survival on the y-axis, juvenile survival is strongly linked to the size of the pups when they wean, so how much energy they got from their mothers. So the bigger you were, the better your chances of survival. Except for this intriguing result that if you're too big, it's actually not so good. Your survival rate drops off. And we suspect that's probably because these guys are so fat that they don't have, they're overly buoyant basically, and it's really difficult for them to get underwater and start swimming. So you read about super weaners, and they're fascinating creatures, but it's probably not a great survival strategy for these guys. So, bigger pups live longer. So this is some work by my student Sophia, who's down here somewhere. She was able to demonstrate that the juvenile survival is also linked to ocean conditions. So what's happening out in the ocean is being translated into how the juveniles are surviving. And the figure on the left there just shows her survival estimates for the two periods of time that we had the branding data, the 50s and the 90s. And the most interesting figure is the one on the right-hand side, which shows that the survival of juveniles is related to the southern annular mode. Now, the southern annular mode it's a climate index that reflects very large-scale changes in the weather in the Southern Ocean. Effectively, it just measures a band of strong westerly winds that can move north and south through the Southern Ocean. So when the southern annular mode is positive, those strong winds are further south, and that has implications for things like the deep mixing layer, for the formation of ice and so forth. So this was the first, not the first, but this was a nice strong indication of the fact that the climate is having an effect on our juveniles and their survival. So we wanted to try and get a bit of a handle on that, so we looked at what was happening onshore in terms of energy transfer from mums to pups. And this figure here just shows the amount of mass gain that pups have during the 3 weeks that they're with their mums. They have a very short lactation period. They've got 3 weeks for mums to pump as much energy as they can into these babies. And you can see from that that the size of the mum, which is the x-axis, is positively related to the amount of energy they can give across to their pups. So bigger mums, bigger pups, better survival. There's some interesting little wrinkles in this one. One is that the male and female pups respond differently to that. So you can see that the red, which is the males, so the females that are biggest actually put disproportionately more energy into their male pups than their female pups. And this is almost certainly a consequence of the fact that they're a highly polygynous species, and that if you can give your male babies a bit of a boost up, that will improve your genetic outputs if they become a beachmaster, because a beachmaster will probably have hundreds of pups. So we looked at the role of environment in this relationship as well. And in this figure here, we've taken we ran some simple statistical models basically, and what I'm presenting here is just comparing two scenarios. So the red scenario is what you might want to think about as good years. So that's when the southern antelope mode is high. Remember that had the effect of increasing survival. And we've got low sea ice, and the blue, we've got low salmon, high sea ice, so that's a bad year. And you can see that if you're a small female, the conditions have a really profound effect on how much energy you can gain. So if you're a small female, you can really increase your ability to make good babies in a good year, but that disappears for the bigger animals, so they sort of converge. So what we're seeing here is there's quite a nuanced relationship between the environment through pup size and eventual recruitment into the population. So the other really important question we had was, where do they go? When we started this work in the 1980s, this map is all we knew about where they went. And you'll notice there's no at-sea information at all. This is really just a map showing the breeding locations of the seals. Once they left the beach, we had no idea. It was a complete mystery. So one of the things about being a marine mammal biologist, particularly in the Southern Ocean, it means that you're an early adopter of new technologies that are going to help you answer some of these questions that you've been struggling with. So in 1986, we put our first elephant seal tag out at Macquarie Island, and the photo there has David Slip and Rupert Woods, who did quite a lot of work on seals back in those days. So this was the first satellite tag we'd ever deployed. It was an interesting tag for all the wrong reasons. The seal stayed on the beach and we got a couple of uplinks, so we knew it was on Macquarie Island. It went to sea and it promptly stopped transmitting. About 3 years later, there was a trawler trawling off the shores of Macquarie Island and it dredged the tag up. And the tag now lives at the museum in Hobart. And I don't know what makes me feel older, being here giving a plenary or having some of my work displayed in a museum. Okay, but we did get the hang of it eventually. And this is the result of 50 tracks of female southern elephant seals from Macquarie Island. Now the colours there are just representing areas that they use a lot and areas that they don't use much. So if it's a blue area, it means that the cells in those areas had a very short residence time. The animals just swam through them, so they're areas of transit. And if it's red, they spend a lot of time there, and these are areas that we could think about as being core foraging areas. So if you look at that map, you can see that those seals from Macquarie Island, and this is just from Macquarie Island, are pretty much spread right across half of Antarctica. They're dispersed incredibly widely. They use a variety of habitats from the deep ocean, from the subantarctic all the way down to the Antarctic continental shelf. And the little black wiggly lines there are the maximum ice extent over, I can't remember, it's probably about a 15-year period. Each line is the maximum ice extent for an individual year. So you can see quite clearly that the areas that the animals are using a lot, those bright red areas, are well within the Antarctic pack ice. And I gave a talk at some marine mammal meeting, I can't remember when, where I was making the case that elephant seals should be regarded as the fourth pack ice seal, along with craybeaters, Ross seals, and Weddell seals. The other thing to note from that figure is that those little black wiggly lines are very variable. So we have some years with lots of ice, some years with relatively little ice. And we saw from some of the earlier analysis that seems to be feeding into some of the things that we're seeing. The other question was, what do they eat? Now, probably not many of you have tried this, but I've tried stomach flushing elephant seals at Macquarie Island and at Kerguelen, and it's the singularly most unrewarding job I've ever done. It takes a lot of work to get them to sleep, to get the stomach pumping and all that stuff. And all you get, if you're lucky, is a handful of highly digested little squid beaks. And that's, of course, because these animals are dispersing incredibly widely, as we've seen. They're travelling thousands of kilometres, and by the time they come back, they've digested whatever food they had, or whatever food they have in their stomach would have been from a very local area anyway. So again, adopting new technologies, we use things like stable isotopes, which is the figure on the left, and fatty acids, which is the figure on the right. Now, it's probably a bit hard to see there, but on the left, the stable isotope figure, we've got three little grey dots, which are the stable isotope signatures from 1, 2, and 3-year-old elephant seals. And we've plotted that up against other predators that the seals might have ecological similarities to. And just to make it easy for you, they are most similar to king penguins. Now, king penguins, we know, are another deep-diving, mesopelagic feeding species. So we're fairly confident that elephant seals are sort of broadly within that niche. They're feeding on mesopelagic prey. They're feeding quite deep in the water column, 400 metres or so. We also used fatty acids to try and get a bit more nuance from that. So every seal that we put a tag on, a satellite tag on, also had a blubber sample taken. So we were able to spatially allocate diet based on where the seals had been. And so the figure here just shows the results of discriminant function analysis of those samples. The little red square at the top is where squid sit in that analysis, and the little purple spot down the bottom is where fish sit. And so the seals obviously are falling somewhere in between there. So anything to the left of that little black line, they're feeding predominantly on fish. Anything to the right, they're feeding predominantly on squid. And I think the interesting things from this figure is that, it looks like they're feeding predominantly on fish, but squid still play a big role in what they're doing. But the thing that's really striking, I think, is that there are these regional differences. So the animals that are in the Ross Sea, which are the... I can't read it from here. The Ross Sea seals are doing slightly different things to the seals from the East Antarctic, Australian East Antarctic area and so forth. And there's also variation through time, so it varies from year to year. So we started to get a bit of a handle on where these seals fit in terms of the trophic webs of the area. So, where are we 40 years on? Now, Sophia asked me to make this talk inspiring. I'm not sure this figure fits that bill, because 40 years on, the population is still declining at pretty much the same rate. So the red dots there are all the counts we've done at Macquarie Island since we started this work in the 1980s. And you can see that it's still going down. It's still declining, despite 40 years of research, a large number of PhDs, and a large number of papers. So you might think that's not particularly inspiring, and it's in fact a little bit depressing. But I see it in a different way. I see this just as a nice illustration of how science actually works. Science rarely delivers those happy stories where you get hallelujah moments and things all fall into place. Science is much more slow, methodical, piecing together stories. And in this case, we've learned an awful lot about these guys. And we can rule some things out, and I think that's important. We can rule out fisheries interactions for a start, because the fisheries in the areas where the seals are very restricted, they're very small, and there's almost no overlap with where the seals are going. The seals are using massive areas. So we're pretty confident that they're not interacting directly with any fisheries in the area. We do know that smaller weaners, a reflection of lower female foraging success, leads to reduced survival and recruitment. And we're pretty confident that that is somehow linked to environmental change, be it through sea ice, southern annular mode, or in some of these other climate indices. But this really raises an important point, because these indices are very blunt tools. I mentioned that the southern annular mode is to do with wind strengths moving up and down through the Southern Ocean. Now, seals don't eat wind, so there's some linkages there that we don't understand. And so the problem is that we're not measuring the environmental parameters at the right scale. We need to measure it at the scale which is pertinent to the animals. And I think without that sort of information, coming up with policy levers that we can pull to try and make a difference to what the seals are doing is going to be very difficult. So we still don't know enough. And I guess my other lessons are that the Southern Ocean is hard. It's a hard place to do this kind of work. We have very limited windows and access to what's happening in the ecosystem. And the third lesson is an obvious one. Long-term monitoring is really fundamental for us to keep tabs on what's happening down there. Okay, so we're going to shift gears a little bit. I'm going to talk now about some more positive things about what we can do in the Southern Ocean. And the first one comes back to that point that I made, that we're not measuring the ocean at the right scales for these animals. We really need to be measuring it at the scale of the animals themselves. Now, thanks to wildlife computers back in the day and the fabulous insights of Roger Hill, when Roger Hill, we ordered a bunch of dive recorders early on, and Roger said, well, it doesn't cost much. I might just put a temperature sensor on these things as well. You might never use it, but you know, it's there if you want it. And that proved to be remarkably insightful. When we got the tags back and we started looking at the weather data, at the temperature data, we found really strong signals about the structure of the water column. And it was pretty exciting. And those of you that are familiar with oceanographic data will realise that we knew so little in those days that we plotted these figures the wrong way around. You should have them the other way around. But we were still excited by what we saw then. And that work then led to a really productive collaboration that we've had in Australia with our French colleagues at the CNRS in France, and particularly Christophe Guinet. So we started a collaboration with them in 2004, so 20 years. We had our 20-year celebration earlier this year. And this program really hinged on using the Sea Mammal Research Unit CTD tags, which I'm sure you've all gone and had a look at the SMRU stand, because they're just phenomenal little instruments. They've been the backbone of this work for 20 years. Now, I don't know how many of you have picked up on the mistake that I picked up on just when I was reading through this talk just before I came on, but that is not a Weddell seal. That's an elephant seal. I had planned to put a Weddell seal in because I wanted to show that we're not just elephant seal biologists, but we work on all sorts of things, but I forgot to do that, so it's an elephant seal. The thing about these tags is that they are giving us really high quality oceanographic data, high enough quality for the oceanographers to take it seriously, and they can use these data for their own research purposes. And they produce up to 4 CTD profiles a day. So when your seals are at sea for 8 months, that's an awful lot of data that we're getting in. So this is just an early representation of the some of the work that we did. This is from Jean-Benoit Charasson. The figure on the left just shows all the ocean data at 200 metres that were available through conventional oceanographic means at the time. And the figure on the right is all the data we collected from elephant seals at that same time. And the really important thing to note are the red dots. They're CTD profiles that came from within the Antarctic pack ice. The usual oceanographic tools like Argo floats don't perform well in the pack ice, but elephant seals love it. So we were basically opening up a whole new window of ocean observing for the oceanographers. Just going to let this little figure run, because as I mentioned, we've been doing this work for 20 years now. And in that time, we've now deployed 850 CTD profilers from Kerguelen and from various other places. And this little figure is just like a historical compilation of those data. The number in the middle is the number of tags that have been deployed at that time. And the little red dots are elephant seals, and the little blue dots, which you see flashing in and out, are Weddell seals. So this is the longest and largest continuous tracking study in the Southern Ocean. It's been an amazingly productive collaboration, and we're really proud of the fact that we've been doing this work for so long. Much as I love watching that, I'll kill it and we'll move on. So the work has, that's a Weddell seal. I told you we worked on Weddell seals. So this work has produced up to 165 publications to date. We've had up to 20 PhD students working on the data in various ways, collected over 800,000 CTD profiles, and over 1 million dive profiles. So an incredibly rich data stream. So I'm not going to dwell on this. These are just examples of some of the many papers that have been published from this work. But I will, I'll stop on this one, because this shows some recent work that's been going on using fluorometry sensors that have been put on these tags. So we're no longer just measuring physical parameters, we're now starting to measure some biological parameters, and that's opening up a whole new world for us. So the seals are now providing 70% of all the CTD profiles that are collected south of 60 degrees. So more than half of the data that oceanographers have available to them to understand what's happening in the Southern Ocean is coming from seals. It's a really nice example of how you get sort of unexpected consequences of going down particular paths. We went down this road to try and work out what the seals were doing, and we end up, I guess it's kind of conservation by stealth. We're actually helping physical oceanographers understand the marine environment in ways that they couldn't do without us. So it's a really satisfying outcome. The oceanographers have used these data to demonstrate the critical role, to understanding the role of dense shelf water production, which is really important in the overturning circulation of the global ocean circulation. So making really sort of fundamental insights. And I did just want to, I see the time's moving along, but I did just want to talk this one, because this is one of my favourite things that we've been able to get from these elephant seal data. And this is, we're remapping the bathymetry of the Antarctic continental shelf. We've got so many dives now on the shelf, we can look at our diving data and see where they disagree with what the current understanding of the bathymetry is. So in this figure, anywhere that's red, that means that our seals are diving deeper than the present understanding of the ocean floor is. And that's super important. You probably can't really see it on this figure, but a lot of these red areas are in front of glaciers, and deeper water in front of glaciers has really important implications for the way meltwater is getting into the base of the glaciers and affecting glacial melt rates and the rate of glacial outflow. So again, a really unexpected and happy serendipitous bit of research. So this work has now become recognised by the Global Ocean Observing System, GOOS, which is like the big parent organisation that dictates all of the ocean observing in the world. It was, until very recently, really dominated by physical oceanographers, but just recently, in the last few years, they've actually recognised a program called ANIBOS, which stands for the Animal Born Sensor Network, and this is now an integrated part of the GOOS observing program, and it's really demonstrating how the animals are now an essential part of the global ocean observing system. I'm just looking at the time there. I might skip. This is my favourite. I'm not going to skip it. This is one of my favourite slides. This shows another really serendipitous thing that we can get from seals, and this is the figure on the right shows some diving profiles from elephant seals. This is work that Michelle Tums did back in the day. And it's how we identify what we call drift dives. These are dives when the animal swims down to a couple of 100 metres depth and then just stops dead in the water. And depending on how fat they are, they either sink, or if they're really fat, they'll float. And so we've got a built-in body condition measure of these guys. And the figure on the left is just one of my favourite figures of all time. It just shows, it's a map of where it's good to be an elephant seal. So using this drift dive data, we can now map up where the seals are feeding successfully and where they're not feeding successfully. So this is my fourth lesson. We've got to think outside the box if we're going to try and solve some of these complicated ecological questions. And I'm just going to quickly touch on this because this is a really exciting new development. This is work being led by Christophe Guinet at CNRS. He's basically turning seals into dolphins. He's putting active sonar tags on the seals so that they are effectively becoming dolphins. And this, of course, has a lot of really important implications The first, of course, is that we'll start getting decent audiences at some of our talks at marine mammal conferences. But the other one is that we can now start actually directly measuring the prey fields, and that's going to be a really exciting development. Okay, I'm just going to finish up then by talking about the other really positive thing that we've done recently, and it's a nice illustration of how the work that we do as biologists can actually start having a bit of addressing some of these policy leverages that I talked about, policy levers. And this is a program that I led called the Retrospective Analysis of Antarctic Tracking Data. It was a SCAR initiative. And this was designed to address a key challenge in managing any ecosystem, and that is knowing which parts of that ecosystem you really want to be able to manage or preserve. There's a lot of problems with using single species, sort of, you hear talk of using it like a keystone species as a representative for this kind of work, but it really has a lot of problems. An alternative approach is to assess space in terms of ecological importance. So our idea was that regions that were used by multiple species of top predators that came from a range of different ecological backgrounds, it must indicate regions of high biodiversity and high biomass of lower trophic levels. So by studying the top predators and integrating them together, we could start identifying areas of ecological interest, not just for them, but for the whole trophic system. So this is a figure of the data set that we were able to collect. We had over 40 data contributors. We basically wrote to all of the people that had done any tracking in the Southern Ocean and asked them to share their data with us. And we had about a 99% uptake. Pretty much everybody we asked for data gave us data. We got data from 16 different species, range of bird species, penguin species, seal species, and even a whale species. So that was fantastic. So we had quite a good range of different species. So despite that, I'm just going to quickly go back to that wonderful little colour picture. Oh, it's not going to do it for me. Oh well. Despite this enormous data set, we still didn't have complete global coverage of all the species in all their locations. So we had to use models to try and give us a global overview of what the animals are doing. This figure here just shows the results of some fairly complicated species distribution models that we did for each one of our species. We then took each one of those species maps and combined them, again using some fairly clever statistical analysis, which I won't go onto, to give us maps of the overall habitat importance, which is the lower left figure. The little green area is our map of habitat importance in terms of all the different species that are using it. We then just made some arbitrary decisions about how we might delineate the areas that are the most important, and here we just use the upper decile. And so the big grey figure just shows the regions that we've just proclaimed as being the areas of ecological significance in the Southern Ocean. And you can see that they're scattered right along the Antarctic continental shelf, and there's a big blob in the South Atlantic, and there's another big blob in the South Pacific. We then wanted to see how this might fit into management objectives. So on the map you can see the areas of ecological significance, the green areas, and then we've overlaid on that the pink areas, which are the proposed MPAs within this part of the Southern Ocean, and the brown areas are the areas that are already being proclaimed as MPAs. Now, marine protected areas are a really big deal in CAMLR. It's one of the things they spend most of their lives talking about, trying to establish MPAs, and having information that helps them define and articulate where MPAs should be is really helpful to them. So the figure at the top there just shows that currently, I can't read the figures from here, but something like 27% of what we've identified as areas of ecological importance are covered by current MPAs, and I think it's something like 35% will be covered by MPAs if they implement all their proposed areas. So that's actually pretty good. It's not perfect, but it's actually pretty good. But we were also interested in how things might change with time. So we got all the climate change models, the CMIP models. There's a whole suite of those, and we use those to predict where our areas of ecological significance might be in 100 years' time. And the figure here shows exactly what that looks like. So the blue areas are where there's no change. So those areas will continue to be areas of ecological significance. But where it's green, they're new areas. So these are areas that are not currently areas of ecological significance, but will become areas of ecological significance. And the brown areas are areas where we're going to lose areas of ecological significance with climate change. And the figure on the right just summarises that in a slightly more quantitative way. I think the important things to look at there are the rows labelled Antarctic and subantarctic. You can see that in the Antarctic, we're going to be losing about 10% of our areas of ecological significance. But on the upside is we're actually going to gain some areas of ecological significance in the subantarctic. Yep, this is my last slide, so it's a miracle. So that's my lesson 5. Lesson 5 is that collaborations, and this was a fantastic collaboration, collaborations are vital, and sharing data is fundamentally important. It's something we should all learn to do, is share our data with people that are doing big synthetic studies. So, after a long career studying marine mammals in the Southern Ocean, what have I learned? So you might think that some of these lessons I put up sound a bit sort of trite and a bit simplistic, but I think that in the Southern Ocean, they're actually still true. You know, we don't know enough. It is hard. We do have to think of new ways to do things if we're going to try and make progress. And I also think that this work has shown us that because Antarctica is isolated, as I said right at the start, it means that it hasn't got a lot of the management problems that we've been hearing about at this conference. So it is, to some extent, protected from a lot of the problems that we hear about in the rest of the world. Now, I'm not trying to minimise by any means some of the conservation problems that I haven't talked about. Nick Gales gave an excellent talk this morning about whaling in the Southern Ocean. There's been a lot of work done on albatross bycatch in the Southern Ocean, you know, birds getting caught in the longline fishery. And there's also some excellent work going on with sperm whale depredation. But I do think that in the Southern Ocean, our biggest conservation challenges are ecological in nature. So we're dealing with things that are occurring at ecosystem levels, not at individual species levels. And I think this is probably part of the reason that we didn't make the sort of progress we really wanted with the elephant seals back in those early days. We're kind of asking the wrong question, or looking at it from the wrong way around. Rather than asking about a single species, we should have really been looking at ecosystem level questions. So of course we need people to go out and study individual species. That's the grist that we need for all of this ecological work. But we also need to be able to take those data, combine them, and do some synthetic kind of work. And that's probably the only way that we're going to be able to provide policymakers with the sorts of tools they need to help them make the decisions they need to make. So perhaps the most powerful tool we have for helping the Antarctic Treaty System and CAMLR is basic ecological research to try and come to grips with how these how these systems are working. Now, I know I don't need to say this to anybody in the room, but I'm going to say it. This long-term monitoring it's the keystone, it's the bedrock of everything we do. And everybody in this room that's tried to do long-term monitoring will just roll their eyes and say, of course we know, but it's so hard to keep it going. And it is, but we have to. We have to keep the long-term monitoring programs going, and that means that we have to be continually reinventing the reasons that we do it. But we've got to do it. Okay, so I'll wrap up there. I said at the get-go that Antarctica is a hard place to work, and it is a hard place to work. And probably the main lesson that I would take from having worked in Antarctica all this time is that in Antarctica you don't get anywhere unless you work as part of a team. It's just fundamental to success. And I think we can broaden that idea out to the science that we do. We need to be collaborating and working in much more efficient teams than we are. And I think Nick actually made this point this morning as well. Collaboration is super important. So if any of you know a physical oceanographer, take them out for a beer and convince them that you can actually help them do their work. It's a two-way street. You know, we don't just want information from them, we can give them useful information. And if you're sitting in your lab and you get a request from somebody who's proposing to do a synthetic study and they ask for your data, give it to them. It's fantastic. These collaborations are productive, and having just done one, they're a lot of fun. So go out and do it. So I'll wrap up there, but I will finish off just by thanking a lot of people that have been with me through this 40-year journey. So there's Clive McMahon, Christophe Guinet, Rob Harcourt, Marianne Lee, Dan Costa, and of course Harry Burton. I'd also like to thank the 50-plus postgrad students who've, to a greater or lesser extent, contributed to what I've been talking about today. I'd particularly like to single out and thank the instrumentation group at SMRU, Mike Fedak and his group, and Wildlife Computers, who made the technology that underpin pretty much everything that we've been doing. There's the funding organisations that have helped fund 20 years of tracking work in the Southern Ocean. And I'd particularly like to thank the field workers, who there are probably hundreds of them who have contributed to this work. Now, it's a real privilege to work in the Southern Ocean, but it does come at some cost. You're away from your families for a long period of time. So I would just like to thank all of those folks. So thank you. That's my reward for being the last speaker.
Yeah. Mark has blown out by 10 minutes. We've got time for a couple of questions because this is a plenary and why not? So if you've got a question, I might ask anybody who's got a comment to maybe just save that for having a chat with Mark later. If you've got a specific question, rock on down or up to a microphone. You're on the right.
Hi, thank you so much. You mentioned the importance of long-term monitoring projects in the Southern Ocean, but many of us working in national Antarctic programs are seeing our ship hours and sometimes even whole vessels being cut due to concerns or efforts towards net zero science. What advice would you give to those fighting to maintain their long-term monitoring programs in the face of that difficulty?
Yeah, and I'm not trivialising that as a problem at all. It's a massive problem. But you've really just got to be a bit creative in how you go about justifying the work. And possibly even look outside your own program to try and get things done. So we're a big community, and there's a lot of people working in the Southern Ocean, and so it's possible you can find linkages with other national programs to help fill some of those holes. But yeah, I think it's one of the biggest problems we face is the cutting of our long-term programs. We're going to regret it in a few years' time.
Thank you.
Yeah, Mark, thanks very much for the presentation, but also for all the work. It's you've spent a lot of time away from your own family and what, so thanks. I'm curious about the decline and the Southern Ellies on your side, and what you might attribute that to, and possibilities like a few of them migrate to the other side of the Antarctic, which presumably you'd see coming the other direction as well. But I just wonder if you have any thoughts about why that steady decline has continued for so long.
Yeah, well, if I knew the answer to that, then I would have told you. But I think, as I said, we're pretty sure it's something to do with the change in the environment. And one of the things that I didn't get a chance to talk about is that we know that the individual seals are really highly site-specific, so they'll keep going back to the same location all the time. And it's interesting also that the decline is actually very small. It's only 1% per year. And that equates to about 20 less seals every year on the isthmus at Macquarie Island, which is why people didn't pick up on it. So it's possible that there's a local effect that's affecting just a small component of the population. which is driving the decline. And we suspect it's probably something like that. It could be some change in the sea ice zone, particularly for animals that are using that part of the world.
Yeah, cool, thanks.
So, is there a way that we can help more the seals by detecting them a little more?
Well, yes, I hope so. So, I think my central message is that we just need to learn a lot more about them before we can persuade people that they need to help them more. And so that's why I'm asking everybody in this room to try and do that, try to work together to try and improve what we know about them so we've got better ability to go out and ask for help.
Louise?
Hi, Mark. I just wanted to thank you for bringing seals to the plenary. And along those lines, I have a slightly sarcastic question to ask, which is, what would your top 5 reasons be to get into seals? You know, how to get PhD students and everything to study seals over their lovely cousins, cetaceans?
Well, cetaceans are obviously worthy animals to study, but seals, you can actually collect data on seals. That's a bit mean. I probably won't go down to 5 because I think Simon's going to get the hook and pull me off.
Great answer, Mark. All your talk has been about the southern hemisphere, but I think we should also realize that the northern hemisphere is facing similar things. So I like to dream, but how realistic would it be if you would start a similar work as you've done on the elephant seals and added the Weddell seals, to do that on the northern hemisphere, where actually ice is disappearing, and we will be losing the animals, or at least the sight of the animals. We have no idea where they're going. Have you had any talks about that? Yeah.
And in some ways, we've almost left it too late because things are changing so quickly up in the north. But some of my happiest times have been working up at Svalbard on Arctic seals. It's a wonderful place. And so I've got a lot of colleagues that work up there, and I know that similar things are happening. I mean, the CTD tags that I talked about were actually developed for Arctic animals, and so there's been a lot of that work going on. But I think the central message is still the same. All the people working up there just need to be collaborating and pulling together in a bit more efficient way.
Yeah. And maybe combining seals and cetacean for different reasons, using one or the other, if it would help to understand better what you're looking at.
Well said. I think that's a good idea.
Please join me in thanking Mark Hindell again.